Welcome to the celebration of Gregor Mendel's 200th birthday by the Indian Academy of Sciences. Mendel, the founding father of genetics, was born on 20th July 1822 and he died on 6th January 1884. It is fitting that the Academy is celebrating this bicentennial because it is now the home of the first English language journal in the field, the Journal of Genetics. The Journal of Genetics was founded in 1910 by William Bateson and Reginald Punnett. Subsequently, JBS Haldane became its editor and he brought the journal with him to India in 1957, editing it from here until his death in 1964. Thereafter, it was published by his wife Helen Spurway until her death in 1977. It stopped publication for some years until 1985, when Naomi Mitchison, Haldane's sister and heir to the Haldane estate, gave the Academy permission to revive the journal under the editorship of Professor Sharat Chandra. Now, by chance, I am the editor of the journal, which is why the Academy invited me to chair today's celebration. I am very grateful to it for extending this privilege to me. I have come with the necessary celebratory paraphernalia, got the party hat, which I'll wear. There are balloons behind me, and there's a noisemaker. There. But the real ace up the Academy's sleeve is that its distinguished fellowship includes Professor Vidyanand Nanjundiah. Professor Nanjundiah, better known to many of us as Vidya, is widely acknowledged for his ability to translate the significance of research done by other distinguished scientists into simple language and thus make it accessible to a wider audience. Anyone who's read Vidya's articles will corroborate this statement. Vidya has described the research done by JBS Haldane and by the great Russian geneticist Vavilov. He has written about the pioneering studies on retinoic acid induced regeneration by the distinguished Indian developmental biologist Iqbal Niazi. He has described the studies of another distinguished developmental biologist, John Tyler Bonner, and yet another, C.H. Waddington. Vidya has described the distinguished sleep and circadian rhythm research done by the Madurai Kamraj University academic M.K. Chandrasekharan. He has written on the interactions of the noted evolutionary biologist Ernst Mayr with JBS Haldane. And of course, he has written on the father of evolution, Charles Darwin. So it was only a matter of time when we'd ask Vidya for his thoughts on Gregor Mendel and the experiments that founded genetics. Today is that time. Vidya was born in 1947. He studied physics as an undergrad and also for his PhD, which he earned from the University of Chicago. Subsequently, his focus moved to developmental biology and evolution. I want to give you a sample of Vidya's research. I will mention only one discovery that he and his colleagues made about 10 years back. They found that a cDNA that encodes the ribosomal protein S4 in the cellular slime mold Dictyostelium discoideum could rescue mutations in the cell cycle genes CDC24, CDC42, and BEM1 of the yeast Saccharomyces cerevisiae. This is a fascinating and completely unexpected finding. Maybe by the time we arrive at Mendel's 250th birthday, someone will have an explanation for the basis of this cross kingdom complementation. I was informed that today's lecture is also being webcast in the 200-seater hall of Tyagarajar College of Arts and Science. We expect there are quite a few students tuning in from there. I have promised that the students will get time for a post-lecture interaction with Vidya. So without further ado, I invite Professor Nanjundiah to deliver his talk. Its title is Mendel at 122.
Thank you very much for these kind words to Gadas, and thanks also to the Academy, especially for its secretary, Rene Borges, for having invited me to give this talk. I'll begin with a brief overview of what I intend to say and then plunge into things. The words gene and DNA are are terms of common usage today. And that's due to a number of reasons. One of them being perhaps the increasing use being made of DNA analysis for charting disease, for prenatal genetic counseling, for genetic modification of plants and animals, and who knows, fairly soon, human beings, for tracking one's ancestry, and so forth. All of these developments and many more involving basic biology, we owe to the pioneering work carried out by Gregor Mendel, whose name you heard a short while ago, about 150 years ago. However, Mendel's work would have remained completely unknown to us except for something remarkable which happened in the year 1900. I should say two remarkable things, really. One was the near simultaneous rediscovery of Mendel's findings by three people working independently. That was strange enough. The second remarkable thing was that Mendel's findings had been buried in an inconspicuous journal, not one that many people read. And these three people happened to chance upon that journal, not in the course of their usual reading or research, but through a chain of circumstances, which I won't bother to describe here, but which you can only term accidental. If that had not happened, it's very likely that the name of Mendel would have remained unknown to us today. But since it did happen, we can celebrate that occasion, and therefore, in a sense, Mendel today is both 200 years old and 122 years old. What I'm going to try and do before you is to give an overview sketch of the history of the gene, so to speak, which is to say the history of the concept of the gene. Uh, I will skim over many important aspects simply because of the lack of time. I hope I will remember to touch on the highlights. The aspects of the gene that I intend to cover are initially what Mendel himself told us and his rediscoverers told us, which is the gene as a unit of heredity. And then I'll go on in serial order to touch on the gene as a physical entity, the gene as a chemical entity, the gene as a unit of evolution by natural selection, and a couple of other aspects of the gene that we have learned to appreciate in the meanwhile. My manner of organizing this might give the impression that these different aspects were revealed chronologically, but that's not the case. Even though there will be a rough chronology to what I say, you can see that many of these ideas were developed in overlapping sequences. Ideas of inheritance, ideas of traits running in families, so to speak, must have been around for a very long time. This sketch illustrates something which is famous as the Habsburg lip. This is a peculiar jutting out of the lower lip, and in fact, if you observe closely, of the lower jaw as well, plus characteristic features to the nose. These are features which ran in the royal families of Europe, uh, primarily of Spain, for quite a few years, for a few centuries, in fact. And we know now that That was because of a large extent of intermarrying among close relatives within these families. In addition to things like this, it appears that there are historical records of things like color blindness and hemophilia running in families. I made a bit of an effort to see whether such records were kept in our history, but I was unable to come up with anything. In any case, a practical knowledge of the fact that traits run in families came perhaps to humanity at large, not so much from the observation of human beings as from plants and animals, which started getting domesticated something like 10,000 years ago. And people made use of this fact that traits run within chains of parents and their offspring. Agriculturists and animal breeders, farmers to begin with, made use of this fact to actually select for plants or animals that possessed traits of their liking. However, it was Mendel's work, as we saw, that really set off the scientific study of inheritance. You notice that in this slide, Mendel has two birthdays. That's because even though July 20th is his official birthday, it turns out that he himself celebrated it on the 22nd, and his family members all claimed it to the 22nd. So those of you who want to can celebrate his 200th birth anniversary once again 2 days from now. So Mendel, as I said, initially established our present day notion of the gene as a unit of hereditary transmission. How did he do that? By making use of experiments which he designed with a methodology which should be our envy even today. He chose the organism on which he wanted to carry out his test very carefully. Besides choosing the organism, he, in a sense, prepared the system because he did not work with the organism, the pea plant, as it was available in nature, but by careful breeding, succeeded in reducing all the variation that was there in the plant available from nature, or actually available from seed suppliers. He succeeded in reducing this to a handful of variations in respect of traits which are of interest to him. Then he proceeded to reduce these variations, these differences between plants, to binaries, which is to say, to differences in a single character which were of a yes or no kind, which were of an either or kind. So this was the simplest kind of difference that he could deal with. And having done all of this, he carried out a breeding program in which, interestingly enough, he traced the progress of these characters that he had already pre-chosen in the parental lines. He traced the progress of these characters in the offspring, but he did so by means of counting numbers, by means of trying to see whether there were any regular numerical relations between different numbers of offspring. Now, this entire approach, which is really not all that unknown in the physical sciences and was not unknown even then, uh, was pretty unprecedented in biology and actually continues to be rather unusual in biology unless you get down to biology at the level of, let's say, molecules or subcellular entities. Having adopted this methodology, he chose, as I said, pairs of traits. Here I illustrate just one such pair of traits, which is the color of the flower, which, as you can see here, is either purple or white. And as I said, once again, this is an either or characteristic. The flower is either purple or white. So beginning with two sets of parents, one of which bred completely true for purple and the other bred completely true for white, Mendel proceeded to cross-fertilize them, and then after the first generation to self-fertilize the offspring. By means of this clever experimental design, he uncovered two facts. One, this trait itself seemed to be underpinned by something within the plant which was also of a yes or no nature, which was also a binary trait. It was, as we would say today, it behaved like a two component system. Secondly, this cross fertilization in the second generation showed him that plants which look identical in terms of their flower color nonetheless differed implicitly in terms of something inside them, because some of these plants, when self fertilized, gave rise to other plants with flowers of their own kind, whereas others gave rise to flowers which resemble the color of the grandparent, as you can see from the sketch. Using a number of such separated binary characters, Mendel was able to establish that, first of all, these characters came as dichotomous traits, either or. Secondly, they seem to combine in the offspring in some manner which was reflected in binary proportions in within the offspring. So, for example, in the first generation, you had a 1:1 ratio of white and pink, which is what he began with because of selfing, of course. But in the second generation, when he crossed them, he had, in the first generation, the cross gives you uniformly purple flowers. In the second generation, you get a 1:2:1 ratio of offspring, except that 1:2:1 presents as 3:1. In other words, some of those 1 and 2 of the first 1:2 pair look identical, but the fact that they are not identical can be revealed by self fertilizing them, when they sometimes give rise to the parental type and at other times give rise to the grandparental type. So, what Mendel inferred from these observations, that there was a binary combination of something within these plants which gave rise to these ratios. He tried the same experiment with different sets of characters: flower color, as I said, seed color, seed shape, height of plant, a number of such things. In fact, it turns out he used 7 such characters, found the same results in all of them, and having found those results, proceeded to combine 2 of those characters in one plant by choosing his initial material, and then discovered that these characters continued to be propagated in the progeny in binary proportions, but did so independently. So the seed shape seemed to be somehow governed by elements which acted independently of the flower color, and so on and so forth. So his inference overall was that behind each visible character, there was some single unitary underlying element which could exist in 2 states, and such elements existed for every pair of characters, but they behave independently from pair to pair. These results were presented in the form of 2 talks in 1865, and then published in this journal, whose title is Proceedings of the Natural History Society of Brunn. Brunn is a small town, actually today a medium-sized town in the Czech Republic, very close to the Austrian border. At the time Mendel worked, it was part of the Austro-Hungarian Empire, so his cultural language, as well as the language of publication, was German. But this journal was obscure, even though Mendel circulated offprints of the paper to a number of people. Either it was not read at all, because some offprints with uncut pages have been recovered since then, or it was read but not understood, his whole approach being rather strange to the prevailing approach among botanists or biologists in general. This work was rediscovered in 1900. The people who made the rediscovery were Hugo de Vries, Karl Scorens, and Erich von Schermark. Uh, the story of the rediscovery itself is rather interesting, but I won't go into it here. Their personalities were, to put it mildly, vivid, and they had run-ins with each other, which were not very unlike the sort of run-ins that we've read about a lot in connection with the discovery of the double helical structure of DNA. But we'll have to pass over all of that. The fourth name on the top, that of William Bateson, is of someone who contributed a lot to genetics later, was not involved in the rediscovery himself, but nonetheless deserves quite a bit of credit because he was the one who publicized Mendel's name all over the place. And in fact, it's due to Bateson that we have this summary presentation of what we now call Mendel's laws, and I'll repeat them just to make sure that you've got them in your minds. The first law is that the visible traits that you see, the visible traits that Mendel saw, came in pairs. That, of course, was by choice. These traits or characters, as Mendel called them, came in pairs, and occasionally one member of a pair could mask the presence of the other. And when I say occasionally, I'm really anticipating myself. In Mendel's case, with all his pairs. Secondly, the entities that seem to underpin these traits equipartitioned, and this equipartitioning, this 1:1 partitioning, occurred at the time of fertilization in through what again to use a phrase which came up only later, through the germline. Different sets of pairs were uncorrelated, and to sum up this entire thing, Mendel showed that there were differences in underlying atomistic or unitary entities which existed in pairs within organisms, which were passed down via equipartitioning through the germline, and the differences in these entities correlated with differences in characters. These three so-called laws are called the law of dominance, which is exhibited by this phenomenon of masking, where you say that one trait is dominant to the other, and the second trait is said to be recessive to the first. This equipartitioning is referred to as segregation, and this lack of correlation is referred to as independent assortment. As soon as Bateson had succeeded in his job of propagandizing the Mendelian gene, genetics took off at a very, very rapid pace, and along the way, it acquired its own terminology. Bateson himself, in fact, coined the word genetics. He also coined the word allele for these underlying entities which differed, which existed in the form of pairs and differed in some individuals. Oddly enough, the word gene came somewhat later. It was coined by a person called Johansen, who also coined the words genotype and phenotype, which have become, in the course of time, very important for those who work in genetics. Genotype standing for the sum total of the genetic makeup of an organism, phenotype standing for the sum total of all its traits, all its characters. Strangely enough, one of the first consequences of the widespread acceptance of Mendel's results and of Mendel's so-called laws, almost the first consequence was a deep divide in the community of biologists among what caused evolution. Remember, we are speaking of the year 1900 and following. Darwin's Origin of Species had already been around for over 40 years, and the Darwin-Wallace doctrine of evolution by natural selection had taken deep root in a number of people, not everyone, but in a number of people who had been convinced by its intrinsic plausibility as well as by the numerous pieces of evidence, which circumstantial evidence to be sure, which seemed supporting evidence, which seemed to support the theory, which seemed to buttress the theory. However, the discovery of Mendel's laws caught many people off balance. The reason being, Darwin's theory or the Darwin-Wallace theory rested strongly on variations being smooth, quasi-continuous, and on natural selection acting, in fact, on these quasi-continuous variations. In fact, drastic variations, discrete variations, what you might call finite variations, uh, would most likely be detrimental to evolution by natural selection. This is not something I will get into here, but this is possible to argue reasonably well. So one had the following funny situation develop. On the one hand, there was this bunch of biologists who you could roughly term Darwinians, who said evolution works along the lines we learned from Darwin and Wallace, namely by natural selection. Clearly, for evolution to work, you need an underlying spectrum of variations. We still don't know how these variations arise. Darwin didn't know either. However, we are sure that they cannot arise through these genes of Mendel, because Mendel's genes lead to significant variations, drastic variations. Therefore, the variations are interesting, and they can have nothing, but they can have nothing to do with evolution. On the other hand, there was a second group of biologists, and interestingly, Bateson himself was one of them, as was De Vries, called themselves Mendelians, who said Mendel's results are here for us to see and verify if we want to. So we have to accept them. They show that variations occur in the form of jumps. Why they occur, we still don't know, but that's how they occur. Clearly, they must be the means whereby evolution proceeds. And in fact, they even show that evolution could carry on by these kinds of sudden jumps alone without the need of natural selection. It's curious that T.H. Morgan, who, as we will see in a moment, uh, did by his work more for the Mendelian theory than almost anyone else, initially supported the Mendelian theory precisely because it seemed to run counter to natural selection. Okay, that's the pea plant. So the next phase or avatar, as I've described it in the abstract, of the gene I wish to describe is the gene as a physical entity. Remember, the Mendelian gene was an abstract concept. The notion of the gene as a physical entity, as something that you could grasp, uh, really goes back, or at least the basis of that notion really goes back to a person by the name of Weizmann, who, well before the rediscovery of Mendel, had postulated that a proper understanding of Darwinian evolution, so he was an evolutionist primarily, Weizmann had postulated that a proper understanding of Darwinian evolution, by which he also meant, let us say, a proper understanding of the reasons why Lamarckian evolution cannot work, Weizmann said that for that, it's necessary that whatever is passed down through the reproductive cells from parents to offspring must come from a subset of cells entirely different from the cells that build up the rest of the body. And by entirely different, he meant not just different in a functional sense, which of course they are, but also different in the sense that they are sequestered and somehow not subject to the vagaries of the environment, which the body obviously is subject to. So this is known now as the germline soma distinction, very useful in the case of animals. So Weizmann said there's some special status to this germline. It emerged that there were entities called chromosomes which could be visualized inside the nuclei of the cells of organisms, and these chromosomes seem to be physically passed down through the germline, through the reproductive cells. But the manner of their passing down was strange because these chromosomes existed in every cell of an organism and existed in the form of pairs of elements, pairs of long threads. It appeared that when they were passed down the germline, the entire chromosomal set was divided in two by a precise binary division. And as a result of this division in two, it was also established that the chromosomes which originally came from the father and the chromosomes which originally came from the mother had got partly shuffled, so that this 50-50 distribution through the germ cells was on the whole a distribution between the originally paternal and originally maternal chromosomes, but with a bit of shuffling involved. It didn't take long for people to point out that this behavior of chromosomes was uncannily similar to the behavior of these Mendelian elements, which had just been a hypothesis postulated by Mendel. And people started asking, look, is it possible that these chromosomes themselves have something to do with Mendelian genes? And these speculations gained weight when it was discovered that, in fact, it was possible to associate one trait, and as important a trait as the sex of the organism, with the presence or absence of specific chromosomes. Initially, this was thought to be a quantitative difference between one sex and another, a quantitative difference in the number of copies of a given chromosome, but soon it was established to be a qualitative difference. So there was the chromosome set of one kind which was characteristic of females, and the chromosome set of another kind which was characteristic in males. Initially, this observation was made on insects, but later was widely generalized. So here was one trait, a binary trait, male or female, associated with chromosomes, and the chromosomes themselves seem to pass down during reproduction in the way that you would expect Mendel's genes to be passed down. So the suspicion grew really that the physical basis of the gene somehow lay in chromosomes. The following phase of work was truly explosive. It involved T.H. Morgan, whose name we came across a while ago. It involved the fruit fly Drosophila melanogaster, and it involved breeding experiments, systematic breeding experiments, which finally pinned down this correlation between chromosomes and genes. But they did more than just pin down the correlation between chromosomes and genes. A young student of Morgan's, an undergraduate at the time by the name of Sturtevant, made the observation that Mendel's laws, which posited independent inheritance of different traits, seem to be violated in many cases. And in fact, in these cases where he noticed violations, pairs of traits seem to be associated either more often than expected, meaning more often than the 50-50 expectation, or less often than expected. So something seemed to be going wrong with this law of independent assortment. So to take a specific case, if you expected trait A to be present in one half the offspring, and the element responsible for trait B also to be present in one half of the offspring, you would expect that A and B co-occurred in one half multiplied by one half, which is one fourth of the offspring. But quite often, Sturtevant found that they were there much more frequently than one fourth. And in other cases, as I said, much less frequently. What was one to make of this? It didn't take Sturtevant long to realize that possibly this association of traits with chromosomes and the fact that you found pairs of traits being correlated with each other may simply have had to do with the fact that some traits occurred closer together on the chromosome than other traits. Therefore, when chromosomes broke and rejoined, remember, this breakage and rejoining involves a fragment of a chromosome from one parent with a fragment of a chromosome from the other parent. When chromosomes broke and rejoined, it was likely that trait elements which were far apart might have suffered a break in between, might have got separated in between, whereas trait elements which were very close to each other might not have. So, using this extremely simple idea, Sturtevant went on to go one step further, which is to actually map these traits in the form of a surveyor's map, if you wish, along the chromosome. The picture that you see on the screen is a highly figurative representation of that, of course. But what it tells you is that you can actually say now that there's a certain physical distance between two of these Mendelian trait elements, and that physical distance can be measured in terms of the frequency with which pairs of traits co-occur or get separated. Curiously, Correns in his paper had remarked on this observation, but apparently didn't go any further. So now things had reached a point where one knew that genes were associated with chromosomes. One could even say genes are, let's say, sitting here on the chromosome, or at least pairs of genes are sitting this distance apart. So the chromosome theory of heredity, as it began to be called, started gaining weight. And in the course of time, it also became accepted that these genes on chromosomes are not really completely stable, but they can change, and they can change in two respects. One respect is when the gene itself changes, and this is a rare occasion called a mutation, recognized once again by a change in the trait that the gene is responsible, that the gene is correlated with. The second change is not a change in the gene itself, but a change in the company kept by the gene, which is to say that the breakage and shuffling that goes on during germline development causes the sequence of genes and therefore the set of genes in a cell to change from what it was in either parent. Both of which we know are important for determining traits. However, they are different in the following sense that recombination, in addition to not changing the gene per se, is a very frequent event. Mutation is much less frequent by a factor of perhaps 1 in a million or 10 in a million. The culmination of this phase of the life of the gene was the publication of this book, The Mechanism of Mendelian Heredity, by Morgan and his students, one-time students, Stratevant, Muller, and Stratevant, Muller, and Bridges. This book has been called the greatest book ever published in the history of genetics, possibly a fair appraisal of the book. Remains readable to this day and strongly recommended for students. It's available online as well. The book basically summed up the results of roughly 8 years of work, 7 or 8 years of work on Drosophila melanogaster, which seem to set the seal on the gene as something associated with chromosomes and as an entity which went through the sorts of gymnastics that we described a while earlier. And it also set in place the ability for people to carry out, they didn't use the phrase at that time, but to carry out genetic engineering, to devise genetic constructs with which highly specific questions could be posed. And in fact, the publication of this book and the work that led up to it set up genetics, the science of genetics, as a formal and elegant field of research, which perhaps has no other equal certainly in biology. Some remarks on this phase, which we can take up later on if questions come up, which is that sometimes the real Mendelian ratios, the expected Mendelian ratios are not observed. Alleles do not always occur in pairs, but it turns out they can occur in any number, but they exist as pairs, of course, within organisms. Genes do not act independently, but they interact via whatever it is that they do. There are certain genes whose presence actually kills the cell within which the gene lies or kills the organism. They were called lethal factors at one time, were very useful from a practical point of view in genetic research. And finally, and perhaps most confusingly, there were genes which seemed to behave in a helter-skelter fashion, completely disordered fashion, in going from parents to offspring. And it took some time to realize that these were genes not present in the nucleus, but present on certain cytoplasmic organelles. And that particular finding led to something very much later, long in the future, which was the realization that we and so-called higher organisms like us are really made up of an ancient symbiotic union of different kinds of reproductive units. Okay, if there are any comments or questions, I can take them now.
My name is Shubhashri Desikan. I'm a journalist with The Hindu. I write on science and technology.
Yeah.
My question is, you had mentioned earlier that choice of organism was very special to Mendel's method, and these people have done their work on Drosophila. So, how does this compare, and can you give some insight into what you said about the specialness of the choice of organism?
Okay, I'll be very brief. The special thing about the choice of the pea was that it normally fertilizes itself. And
Yes.
due to some curious quirk, the pea flower bud does not open before fertilization has taken place. So the pollen from a pea plant from inside the flower fertilizes its own oocyte, and it's only then that the flower opens. Now, one very important practical consequence of this is that contamination from foreign pollen is almost entirely avoided. That is a problem that breeders have had to contend with practically every other organism that they work with. In the case of Drosophila, the flies were kept in milk bottles, were sequestered from other flies, and so on. Now, it's very difficult to do that kind of thing with plants. Mendel, of course, carried out cross fertilization experiments as well, but they were done painstakingly by artificially teasing the bud open, emasculating the plant, and then introducing foreign pollen, which he had brought from another plant with a thin needle, and putting it, getting it to fertilize this plant. Very painstaking operation. Many of these had to be carried out with a microscope. He ruined his eyesight in the process and so forth. Anyway, in the case of flies, they naturally cross with each other. So there's no question of this business of inbreeding. So this advantage Mendel had of utilizing both self-fertilization and cross-fertilization was not available.
This is so amazing, actually, that he should be so conscious of doing this very cleverly. So,
Yeah, the more you read about Mendel, the more amazed you are. Remember, he was a monk. He had no formal training in botany or in any science. He had studied physics and mathematics, but trained to be a teacher and didn't pass his exams and so forth. That's a different story in itself.
yeah, thank you so much for the clarification.
Now I'll run through these different slides until I come to the last set on which I want to spend some time. So I'll tell you in advance what these contain. Very soon, it turned out that this physical association of genes with chromosomes could be complemented by a chemical association, and the chemical association involved specific chemistry carried out by these genes, and specific to the extent that genes were associated with proteins, with individual genes being associated with individual proteins. But the story actually begins with the discovery of DNA much longer ago, the year of 1869, and that is an interesting story in itself, but it languished, so to speak, without in any way being connected with the story of genes until much later. But I mention it just for the record. Curiously enough, we've been through a plant, the pea plant, and we've been through an animal, the fly Drosophila. There were many other creatures that in the meanwhile had participated in genetics research. Humans entered the scene now, and this was very soon after the rediscovery. As you see, the year was 1902, when a physician, Garrod, came across patients who came to him with a strange symptom, which was that their urine was turning highly dark colored. Now, Garrod again was clearly a man ahead of his time, a person ahead of his time. On looking into this carefully, he discovered that all of these people had parents who were closely related to each other, and something clicked in his mind because he had read Bateson's articles eulogizing Mendel and his work. Remember, I told you Bateson was the preeminent propagandist for genetics. And Garrod seems to have said to himself, look, these people seem to be behaving in the way that Bateson tells me Mendel's pea plants are behaving, because this is exactly what happens when two recessive traits, remember the white color, two recessive traits combine together in one individual, and you suddenly see a trait being expressed by this individual which was not there in any of the parents. He did a bit more study into what was happening and discovered that what these people were lacking in was a specific enzyme, and this correlation with their being offspring of consanguineous marriages was established. He applied to Bateson for his advice, and Bateson said, yes, you seem to be on the right track. Go ahead. So Garrod's paper, which is really, again, a fine piece of scientific research, mentions Bateson, thanks him, and says that, perhaps we have evidence here of a Mendelian trait in human beings. The point for us, however, is the absence of a particular enzymatic activity which led to that trait seem to be related to consanguinity among the parents, therefore seem to be related to something genetic. So here you had a link of a gene with enzymatic activity. Actually, it's curious that the trait that these patients came to see Garrod with was dark colored urine, because color was a very popular trait for many people to work on for two reasons. One, variations in color are easily detected. Two, animals actually come with different colors, and you should think of insects here in particular. And secondly, the chemistry of color production, of pigment production, had been worked out to some extent. At least it was an area in which chemists said they could work. So there was a lot of work which went on possible genetic basis in both animals and plants which led to color variations. And one of the leading workers in this was a person whose name you heard from Durgadas a while earlier, JBS Haldane. But this time, the final clinching evidence came from work done on a fungus. So you have yet another organism entering the picture. And this was done by a pair of people, Beadle and Tatum. What they established was, first of all, something similar to what Garrod had done for his patients, which is that it was likely that the absence of a gene was correlated with the absence of a specific enzymatic activity. But more interestingly, Bateson and Garrod managed to show by rather elegant analysis and reasoning that this correspondence held across the board for specific metabolic pathways. Metabolic pathways, remember, track the food that you eat all the way up to whatever the digestive products are. And these are complex pathways of substrates and products, which later became substrates for some other reaction, and products which become substrates of some other reaction and give rise to products, and so on and so forth. Quite often they're written as linear pathways for simplicity. So Beadle and Tatum managed to show that the defect of a specific enzyme in successive steps of a particular metabolic pathway, essentially a food digestion pathway in the mold Neurospora, could be correlated with a genetic mutation, which they could identify by means of genetic crosses, a la Mendel. So this looked very nice, and it appeared, in fact, that a characteristic gene was in some way responsible for a particular enzyme. An enzyme, of course, is a protein. So the essential link here, as far as we are concerned, is between genes and proteins. Very much later, I warned you already that this is not going to be chronological. Anand Sarabhai and colleagues managed to show, working on something completely different in this case, which was a bacteriophage, they managed to show that not only were different genes correlated with different proteins, but different positions within a gene could be correlated with different positions within a protein. I'm putting this rather crudely, but you get the rough idea. So the link between genes and proteins seem to be strengthened. This, of course, went eventually, even though we are moving back in time, went eventually to the Watson and Crick demonstration of DNA as a molecule made up of defined chemical entities. Therefore, it appeared not strange to think of it participating in chemical reactions. This sequence I will pass through very rapidly. I will simply pick, I think, the last slide of this sequence, which goes to show how the notion of the gene as a unit of natural selection took hold. A number of people working together between, roughly speaking, 1910 to, I would say, perhaps 1935, thereabouts. So again, over a period of time that overlapped with some of the things that we've been talking about, managed to show that you could explain the course of evolution by natural selection by assuming that natural selection led to a change in the properties of organisms, which of course was Darwin and Wallace's postulate, change in the property of organisms via changing their genetic makeup. And they did this purely by means of plausibility arguments. So these were all mathematical treatments. And the plausibility argument went along the following lines. They said, let us assume that the traits of an organism are determined by its genetic makeup, or at least determined to some extent by its genetic makeup. Even more, let us assume that this makeup can be broken down into single gene effects. So each individual gene contributes something to any trait that's of interest to you. Therefore, let us assume that if there is genetic variation in this genetic locus, in other words, if there are alternative genes, or as we would now say, alternative alleles at this locus, these differences contribute to alternative traits. Therefore, natural selection indirectly picks for, to use very, very crude language, I must say now, picks for good genes vis-a-vis not so good genes. And if that is the case, in the course of time, the genetic composition of the population must change. Not only must it must change at a specific rate, given a certain intensity of selection, given a certain strength of selection. This they managed to show. So they managed to show that the kinetics of natural selection could be accounted for plausibly by assuming that genes were at the heart of traits. But they did something else then. They also showed that the end point of natural selection under any one set of circumstances could be reached by a certain set of genes continuing to exist in stable equilibria. So these were two important kinds of problems that were tackled by this group of people known as population geneticists. On the one hand, to track the course of evolution and make it plausible that it worked by natural selection, that is the Darwin-Wallace mechanism. And secondly, to make it plausible that equilibrium ratios of different gene combinations could be eventually reached. One consequence of this situation was that it became embedded in the minds of many people that evolution has to be looked at in terms of genetic change, and that I would think is really the prevailing view today, even though there is an undercurrent of opinion which holds that that is not a very useful point of view to take. But the prevailing view is that genetics, I beg your pardon, what am I saying? That evolution must be measured in terms of changes in gene composition. And in fact, there's even a mantra which says the frequency distribution of different genes in a population is the signature of the population. And therefore, any change in that frequency distribution, in other words, any change in the relative proportions of different genes at different loci, tells you that evolution is taking place. This is a famous example of a genetic equilibrium and therefore temporary evolutionary equilibrium, but I won't go into it in details, but I will pause again just for a moment.
Subhashri has asked a question again. Can you explain the last statement?
Yeah, could you please tell me what you mean?
I meant that you said that the prevailing theory is that evolution is mainly through the modification of genes, right? So what is the other view? I was curious. I didn't know of.
No. Okay, so I'm glad you asked that question because I've given the wrong impression. I didn't say that's the prevailing theory. I said that the prevailing opinion is that the best way to look at evolution is in terms of changing gene frequencies. The best way to look at evolution. Obviously, the question crops up, can evolution take place within a fixed assemblage of gene frequencies? And apparently, in some cases, if evolution in the long term cannot take place, evolution in the short term, change in the short term can perhaps take place. So such people, and by the way, I must confess, I'm one of them, think that one shouldn't put too much weight on the notion that only changes in gene frequency are important for evolution.
There's another question from Medha Rajadaksh whether it would have helped Darwin in his arguments had he known about Mendel's work, and this could have led to an earlier acceptance of his theory by scientific critics.
That's a very interesting question. And I'm tempted to answer no, that Darwin, in retrospect, should feel glad he didn't know about Mendel's work. Because as you remember, the initial response to the rediscovery was that Mendel's work had, I shouldn't say Mendel's work, that the existence of Mendelian genes had disproved the possibility of natural selection. So this work that I've glossed over so rapidly, the gene as an evolutionary unit, had at its heart a very important contribution by R.A. Fisher, who showed that even though genes taken singly underpinned discrete changes, when you looked at genes in the aggregate, meaning when you looked at traits that were liable to be affected by many genes simultaneously, then the changes would indeed become quasi-continuous. And it was Fisher's demonstration of this that finally made it evident to everyone that, in fact, genetics was central to Darwinian theory and not opposed to it.
Yeah, thank you. Thank you. I think that's very clear.
Thanks. So this is really, in some ways, a peculiarly late 20th century notion, the gene as an informational unit. And the reason this notion has come up, and the reason the word informational is in inverted commas, is as follows. First of all, the second point. The word information, of course, has a very technical meaning. And people who work on information theory will probably tell you that it means what's called Shannon-Weaver information. So in some sense, the information in a message is the measure of unpredictability in that message. But that is not the sense in which information is being used here. In genetics, information is used more in the semantic sense, in the sense of something conveying meaning. Now, why is this sense different from the conventional sense of DNA and genes as chemical entities which take part in chemical reactions, and by virtue of these chemical reactions somehow give rise to the production of proteins? Isn't that also information transfer in itself? The answer is no, and to say why, I have a sketch which comes up. It starts with this book by Schrödinger titled What is Life, in which he first raised the possibility that genes do whatever it is that they do, not by means of standard physics and chemistry, but by working in the form of encoded information, in the form of a Morse code. This theory gained ground following the announcement of the double helix structure by Watson and Crick, and after a great deal of work, which culminated in this spectacular discovery made by many people, prominent among them Nirenberg and Khurana. Incidentally, we ought to have celebrated Khurana's birth centenary earlier this year. Anyway, this work of trying to see how you get proteins from DNA, what the underlying chemistry is, culminated in the establishment of what we now call the genetic code, which actually justified Schrödinger's use of the term, meaning that it turns out that genes give rise to proteins because genes are strings of DNA. These strings of DNA can be read off 3 molecular units at a time. The units are symbolically represented here on the left side of each of these columns. The units come in only 4 types: A, T, G, and C, or A, U, G, and C, in a certain sense. They are read off 3 in a time, and amazingly enough, each combination of 3 units corresponds by a rule of correspondence, not by conventional chemistry, corresponds by a rule of correspondence to a particular amino acid. And those amino acids are listed in the right-hand part of each of these 4 columns. So this is the essence of how DNA gives rise to proteins. DNA contains a sequence of nucleotides. This sequence is read off in groups of 3. Each bunch of 3, there are certain exceptions and there are fine points that we don't need to get into here. Each bunch of 3 corresponds to a particular amino acid, but that correspondence is just a correspondence. It's the same kind of rule that you might use, for example, in linguistic correspondence, where you say that a certain pattern of sounds made by you has a certain meaning, which is interpreted by a hearer who knows that language. And this rule of correspondence, to all appearances, is arbitrary, meaning there's no logical reasoning that you can give. People have tried very hard, by the way, and some of them have made interesting advances, but there's no fully satisfactory logical reasoning you can give to show why a particular triplet of nucleic acids gives rise to a particular amino acid, which later goes on to build a protein. To the extent that this rule of correspondence is arbitrary, by the way, the implementation of the rule is via chemistry. So there are certain chemical reactions which take place, which involve the binding of one entity to another entity and so forth, which enable you to go through this sequence. But it is not ordinary chemistry of the transformational type. It's not chemistry of the sort which tells you that if you add an acid to a base, you must get a salt and water. So there's no underlying chemical principle with the help of which you can predict this code. So it's in that sense that DNA has come to be thought of as an informational entity. We've come to the realization now that the way genes work involves many things, building bodies if you wish, many things which involve gene-mediated reactions as well as physical effects without changing the primary genetic constitution of the organism. And this is a fairly active field of study today, which goes by the name of epigenetics. I just bring it in to throw the word at you. So this is the question that I asked already. Where did this genetic code come from? Is it arbitrary? Is it a frozen accident? Or, as some people contend for interesting reasons that I can't explore here either, is it a product of evolution by natural selection? Or finally, is there a logic underlying it all and we just don't understand the physics and chemistry involved? We are now living in a conventional picture of the gene, which I have been sketching to you so far, has started getting unsettled. The picture that we have sketched, remember, is that the gene consists of a DNA sequence of the chromosome. The DNA sequence read continuously corresponds to a sequence of amino acids via the genetic code, therefore to a protein. How much of a protein is made, where it is made, and when it is made are all regulated by the activities of other genes which make other proteins. This was the conventional picture of the gene until certainly 30 or 40 years ago. That has got destabilized by a number of findings, again, some of which had occurred much earlier. One of the most dramatic of them was that genes can be unstable. So something like mutations seems to occur during the lifetime of organisms and gives rise to bizarre variations, which we now understand in many cases as being the results of genes actually jumping from place to place, and by virtue of jumping, sometimes integrating themselves into the middle of other genes, which they consequently mutate. Another finding which goes back a long time ago is that the link between genes and proteins, and therefore link between genes and traits, is not as simple as we have said or pretended so far, because environmental factors play a huge role, and the same set of genes, i.e., in the same organism, can give rise to drastically different traits depending on the environment. This is referred to sometimes as the plasticity of the phenotype, again, a very active area of study. Genes can be split. This came as a huge shock. So you can actually have gaps in the middle of a gene where, sure enough, you do have molecules. The DNA molecule is continuous, but those molecules may or may not have anything to do with the message of the gene in the sense of the protein that's formed later. And in fact, there's a whole complex series of activities which goes by the name of splicing, in the course of which these intervening stretches of DNA are thrown out. One gene can give rise to many proteins. It's a consequence of what I said just now. If a gene has several gaps within it, splicing can give rise to different combinations of coding sequences and therefore to different proteins. So you no longer have a 1:1 mapping between genes and proteins. The old Mendelian idea that a single gene could be correlated with a single trait is pretty much out of the window now. Very surprisingly, but also anticipated by some people, DNA sequences need not code for a need not code for a protein at all. So you can have genes which don't code for proteins. In fact, something like 90% of the genes in our body do not code for proteins. There's a fair amount of debate as to whether these genes that do not code for proteins nonetheless perform some useful function for the organism. In some cases, we know the answer is yes, because these genes, it turns out, give rise to an RNA sequence. I won't use the word code for an RNA sequence because going from DNA to RNA is straightforward chemistry, doesn't involve coding. So there are some genes which give rise to an RNA sequence, which turns out to be useful in rather interesting ways. And there's a fair amount of work going on to that. Nonetheless, it appears that there's a fairly substantial amount of DNA in our bodies which may well not be functionally useful. Secondly, and we've already come across this many times, genes do not act singly, but they act in concert. And not only do they act in concert, they seem to act as parts of what you might call consortia. So the link between genes and traits gets very, very complicated. So this buzz of straight lines and various other circles and triangles and so forth is meant to illustrate a specific case in which the products of one gene interact with the products of another gene. So these edges in this diagram indicate interactions, and you can see they're very common. And I simply picked up this cartoon from a paper on genes, which are believed to be involved in cancers of various sort in human beings. And you can see immediately that any one of these cancers is is potentially capable of being affected by any one of a large number of genes. Fact holds true in the case of any trait you wish to look at. So, in short, the genotype phenotype relationship is very, very complicated, which makes you go back once again to Mendel and congratulate him for having so carefully worked in preparing his system and avoiding all of these complications. I'll just mention two of the complications that are listed in this slide, which which we've not touched on so far. One is what I've called stochasticity. It appears that certain genes work in a yes or no fashion, meaning in a fashion that cannot be predicted deterministically, and they give rise to what you might, in a general sense, call noise at the level of gene expression, which also has an interesting consequence in those cases that have been studied. And then there are other genes that have different effects depending on which parent they come from. This is the last part, and I think I have a single slide here. Next slide, please, where we are today. We have the conventional picture of gene action, which holds good to a large extent, summarized in the form of this central dogma by Crick. And at the same time, we have themes that we still need to wrestle with. And I've just summarized these themes. Some of them we've come to accept, others continue to be puzzles, but they remain underpinning our picture of genes and how genes interact and interrelate with organisms. And the themes are those of discreteness versus continuity, which the universal nature of genes, with statistical regularities and accidents as being important factors. Two puzzles that we've still not sorted out are where did genes come from? Where did this whole business of the genetic code and DNA as the genetic material, even if you go back one step, RNA as the genetic material come from? Which probably goes back to something like the origin of life and is really not tackleable at present. And there are other puzzles where you have replicating entities which seem to resemble genes, but are very clearly something very different, and we still don't know how to deal with them. As with any other account, I end with some breaking news, except that this is not really breaking, except for the last one of them. And the breaking news is this. This is some years old, by the way. The pea genome has been sequenced finally, and it turns out that it's larger than our genome, meaning the pea plant has more genes than we have. This is true both in terms of the DNA content of the plant as well as in terms of the number of proteins made by the pea. And now you can think of what to make of that. Secondly, we now know what the genes were that Mendel worked with. That's a point of some interest, and what roles those genes play. Needless to say, they don't indicate an obvious link between genes and traits. Finally, and this is really curious, and this is the only part of what I have to say which is really breaking news. As we are chatting just now, it turns out that there's a Mendel bash going on in Brno, where Mendel did his work. And I discovered to my surprise that one of the talks is going to be on what I think I've got the title approximately right, is going to be on what they call an anthropological and genetic analysis of Mendel's remains. That's a somewhat grisly note on which to end, but I do so here and thank you all.
I'll just ask a relatively, simple question that sort of has been bugging me for some years. People have seen twins for ever since people have been having children. So that means forever. They have probably had twins, and yet they notice that sometimes you get boy-girl twin pairs, and you get boy-boy and girl-girl, and sometimes they are more identical than others. So, was there any scientific study of twins prior to the all of this, prior to Mendel? Because, or were they just sort of not covered in the literature at all?
You know, Professor Sharat Chandra asked me this very question a long time ago, probably 30 or 40 years ago. And at that time, my answer was that he should know. Now I'm tempted to say you should know. I've tried to look at it a little bit, then at the time, and then occasionally now and then. I've not come across anything that involved a systematic analysis of the nature of twins. Certainly not in the older literature. There's a huge amount of twin studies, of course, as you know, which went on in the middle and late 1900s and undoubtedly is going on even now in all sorts of fields. But I've not come across any systematic study. Now, Professor Sharat Chandra posed another question, which I think was also embedded in yours, which is, did people recognize that there were two kinds of twins?
Yes. I mean, clearly the boy-girl twin pairs can't be identical, so they would have had to have an explanation for those kinds, but they occur at a certain frequency, and the boy-boy, girl-girl twin pairs, obviously, because they also include the monozygotic twins, they must be occurring at a higher frequency, and that is simple arithmetic that now explains it all. So whether all these smart people like Gauss and Euler and so on, who need so many other things.
I would love to know, especially this distinction between monozygotic and dizygotic twins of the same sex.
Yeah, because it's at the heart of genetics ultimately.
Yes.
Yeah.
And regarding this twin, that in Tamil Nadu and Kerala, there are many villages wherein almost every family has twins.
I've heard of that, yes.
Yeah, that will be I'm sure that many of you know that the duck-billed platypus, mammal, they are quadruplets and they are all identicals. So that would tell the mechanism of twinning and so on. Yeah, that is one comment. And leave apart.
Well, I'm tempted to interrupt briefly
Yeah.
to add to say that armadillo as identical octuplets. So it's one step ahead
Okay.
of the platypus.
Yeah.
But that really is traced down to what you might call a developmental problem, not so much a genetic problem, even though undoubtedly there
Yeah.
are genes behind that too.
Right. So then, for the purpose of the maybe students hearing this, these are the historical development of identification of genes and so on. Now, as you highlighted the overall differences, epistatic differences and so on, there are a few things in the market available in plenty, confusing the commoner. One is gene therapy for Alzheimer. Of course, Bangalore is a place which offers that. And then you can also tell something about the CRISPR. And as for India is concerned, once again, one of the major stem cells. This is little away from the Mendel, but still, these are the developments which people amazed well through that. What is your comment on this as far India is concerned? Western world is different.
Well, I really am in no position to offer an informed comment on what you've said. There are all sorts of brave new world
Yeah.
scenarios that may lie ahead of us. This whole area of eugenics, which is the engineered alteration of the genetic composition of a population, has certainly been talked about for a long time. And I'm scared to think that it might be gaining a foothold in our country as well. And of course, many of the newer technologies seem to offer a ready opportunity of actually tinkering with human genes in ways that are believed to be subjectively for the good. It's just a very worrisome prospect. I don't want to say more about that.